We are recording. So, what is your first question?
Just give me a second. I'm like waking up.
Okay, that is fine. While you are doing that, I will go to my Google Classroom and get things ready for you to say this study guide or that study guide. I'll be ready to go.
All right.
Well, one step closer to being ready to go. So also while you are doing that, I'll go ahead and remind people, though I have a feeling anybody who's watching this video will not need reminded, but just in case, the exam is again, it'll be like it usually is for those people taking it online. It's scheduled to open at 8 a.m. tomorrow, meaning I've already written it, or I will have written it by then, and I put a little thing in the computer and it says, schedule it for 8 o'clock. What that means is as soon as you see it, even if it's earlier than 8, you can start it. And to remind you, and I got a great question yesterday, where it pops up. There's two places you're going to see it. You're either going to see it in your email, just like you do whenever I make an announcement on Google Classroom, you get an email. You'll either see it there, or you'll see it on the Google Classroom itself. And let's take a look at that really quickly. The only problem is if you're waiting for it on Google Classroom, there we go. You should be able to see that. Kenneth, can you see my screen where I'm looking at Google Classroom?
Yes.
Okay. If you're waiting for it here, this is where it would pop up to, right? It'll probably show up right above exam 2. If you're just sitting here waiting, it's not going to show up. The only way it's going to show up is if you go to this top left and keep hitting the refresh button, right? That's the only way it's going to pop up right here. It won't just show up if you're just watching a static screen. So if you're doing this for some reason, make sure you're constantly hitting that refresh button, which is why, in my opinion, you should probably, instead of doing that, you should probably just keep an eye on your email and maybe even hit the refresh button on that too. That way you can get the exam as soon as it comes up, because again, no matter what time it starts or opens, you can start it whenever you want, but you have to submit. You have to hit the submit button at 8:50 a.m. You will lose 10 points for every minute that you are late, right? So if you're 5 minutes late, you're going to lose 50 points. So even if you were to score 100 on your exam, if you would submit it 5 minutes late, that is, you're going to get a 50, a low, or not a low F, but an F. So make sure you submit it on time. As I said earlier, if you have any technical issues where you have to start late, and someone said that last time too, man, I feel so bad. I just found out yesterday that somebody had to start exam 2 late. If something happens and you can't start on time, just don't take the exam. Just stop what you're doing. Contact me and we'll figure something out, because I'd rather you take the exam a little bit late. You're not going to get penalized if you have to take it tomorrow afternoon as opposed to tomorrow morning. If there's any technical issues, just stop what you're doing. Do not open the exam and contact me. So anyway, you ready now, Kenneth?
Yes.
All right. What's your first question?
Okay. So on the Chapter 9 study guide,
Okay.
on question 38, it says red-green color blindness is inherited as a sex-linked recessive trait. The gene is found on the X chromosome. Can a man with normal color vision father a daughter who is red-green colorblind?
Good. Okay. So what we're going to do here then to show you this is I am going to have to do, well, first of all, let me find the question first so we can put it on the screen. There we go. Red-green color blindness is an inherited sex-linked recessive trait. Let's break this down sentence by sentence. First of all, we know now that we're dealing with, hold on, there we go. We are dealing with a sex-linked trait, which means it's on a sex chromosome, right? It's either on the X or it's on the Y. It's not on any of the other chromosomes. It's on the X or the Y, right? That's what it means by sex-linked. Recessive trait. Now, you hopefully are already familiar with recessive. If you're not, you should. I'm not necessarily talking to you, O'Brien. I'm talking to anybody watching the video. If you're not familiar with recessive trait the day before the exam, you have a lot of studying to do. But when we're talking about a recessive trait on a sex-linked gene, it's slightly different. Generally speaking, for a recessive trait, you have to have both recessive alleles, right? Because if you have one dominant allele, you're going to have the dominant phenotype. So for most recessive traits, you need 2 recessive alleles. However, when you're talking about sex-linked traits and you're talking about a male, right? A male has an X chromosome and a Y chromosome. They don't, right? They don't have two X chromosomes. They don't have two Y. They only have one of each. Therefore, if a male has a recessive allele on the X chromosome, then that's enough for them to have the recessive trait. Because, yes, they only have one allele, and normally you need two. But a better way of looking at it is saying that 100% of their alleles are the recessive alleles. Does that make sense?
Yes.
Okay. So, yes, they don't have two copies of it, but that's okay because 100% of their alleles are recessive. And the same thing can be said with all that other stuff we talked about, all the normal stuff like chin dimples and blue eyes, right? Blue eyes, if you have blue eyes, yes, technically that's because you have two recessive alleles. But a better way of saying it in this context is 100% of your alleles are recessive. So anyway, that's what we're dealing with when we're dealing with red-green color blindness. And then your book points out that the gene is found on the X chromosome. So again, I've already brought that up, right? So for a male, if it's on the X chromosome and they have the one recessive allele, 100% of their alleles are recessive, therefore they have the trait. So can a man with normal color vision father a daughter or who have a kid who's a female who is colorblind, right? So let's hopefully this works for me and I can show you the whiteboard here. These work. There we go. All right. So can you see the whiteboard that I just popped up?
You just shared something.
Okay.
It's a link.
Yeah, go ahead and open the link if you don't mind. Or if you can see it now, can you see where I'm working with it? Even if you haven't opened the link, that should be what's visible. I've shared my screen, so you should be able to see what's up here.
Yeah, I can see it.
Perfect. Okay. So here we go. Really quickly. Well, not really quickly. I'm going to try to make this as quick as possible. This is a male we're talking about. So he has an X chromosome and a Y chromosome. The X chromosome is the one we're looking at because that's the one where this allele would be found, right? If we're talking about red-green color blindness, this is where it would be found. Now, in order for this male to have it, his genotype would have to be X little n Y, right? Because he would have to have that recessive allele for him to have the color blindness. However, the question specifically says that this male is not colorblind. Is that correct? Can you see it? Because I can't see it right now. I'm on my whiteboard. Am I correct in saying that, O'Brien?
Yeah.
Okay. So he is X big N Y. This man does not have color blindness, so this is his genotype. Now, let's look at a Punnett Square. Again, he's X big N Y. Actually, just to save time and to really get you guys thinking the right way, forget a Punnett Square. We know that he's not colorblind. We know this is his genotype. Let me erase this. Now, males only have one X chromosome, right? So again, they only need one of the recessive alleles to have the condition. Because if they have one, then 100% of their alleles are recessive. Now, if we look at a female, right, this is a female. She has two X chromosomes. So what happens if she has, I don't know, a big N and a small n? Would she be colorblind or not?
Yes.
She would not be. Because this is a recessive trait, right? And just like anything else, to have a recessive trait, 100% of your alleles have to be recessive. Only one of her alleles in this situation, if we're saying she's heterozygous, only one of her alleles is going to be recessive. The other one, because she's a heterozygote, would be dominant. Therefore, she would not show the recessive trait. You get that?
Yeah.
Okay, it's a very important concept, not just for this question, just in general. It's a very important concept. So a female, unlike a male, has to have two copies of this of this recessive allele, right? One won't do it because that would only be 50% of her alleles. She needs 2 copies of it. Therefore, without even doing a Punnett Square, what we know, we're going back to the question here, this man in question, this guy right here, with normal vision, his X chromosome is a big N. So we know no matter what, it doesn't even matter what the female is bringing to the table. At the very least, this man is giving his daughter an X with a big N. So the other one could be a little n, the other one could be a big N. It doesn't matter. There is no way for this man to have a daughter who is colorblind because he is giving her a dominant allele, right? So the answer, in short, is no. There's no way of happening. And if I remember correctly, I like the way your textbook wrote that question because it says something like, I think it says something like, no, he cannot unless there's a mutation. Isn't that an option? Unless there's a mutation?
Yeah.
Yeah, and that's the answer, right? And I showed you why, because you cannot, a man who's not colorblind only has the one allele, and he has the one dominant allele. So that is the one thing that he's giving to his daughter. Therefore, it does not matter what the mother's bringing to the table. She will have at least one dominant allele. Therefore, she will not have a recessive trait. And again, like the question mentions, unless there's a mutation, right? If a male, if that male that we're talking about is, in his testes, he's producing sperm, right? He's going through meiosis. So if there's a mistake when he takes his normal allele, and in the process of making sperm, there's a mistake in that process, and then that normal allele has a mutation, and then it turns into an allele where it is recessive, then she would have it. So there you go. Great question. I'm glad you asked that one because that was more than a here's the answer. That was one that I could actually explain. So good question. Also, sorry, this is a good time to point out, and I know some people are going to hate to hear this because they might not agree, but generally speaking, my exams are not that hard. And hear me out. What I mean by that is in my opinion, there's 2 different types of exams. And generally speaking, Biology 101 is the type where you just have to remember stuff. Is it this or is it that? What's the definition of this? What are the stages of this? In what order does this happen, right? That's just memorizing stuff. That's it, just memorization. Harder stuff is actually having to figure stuff out, like a math class. I think that's why people think math is so hard. You can memorize a formula all day, but notice a test on an algebra exam doesn't say, what is the formula for this? What is the formula for that? No, you actually have to apply that to do and work out problems. So most of my questions in this Bio 101 class are just memorized stuff. And O'Brien, you picked a great question because that was one of the few exceptions. There was nothing to memorize there. That answer cannot be found in your book. You had to apply the knowledge that I've given you and that the book has given you to figure something out. So keep that in mind. There, as always, most of the questions are going to be easy in that I'm just requiring you to regurgitate some information. But some of them, a few of them, will be hard, where like that one that O'Brien just showed us, because it requires you to put your knowledge to use and figure something out. And if you take a late exam, that's exactly what you're going to get. The later you take the exam, the more of those kind of hard questions you're going to get. Because, you, in theory, if you took an exam later than your classmates, you would have had more time to study for it. So anyway, O'Brien, what is your next question?
Give me a second.
Okay.
Chapter 10.
Okay. Let's look at chapter 10. While we'll just open it up here. All right, chapter 10. What's the question? What's number?
40. HIV, human immunodeficiency virus, must use its own blank to reproduce.
Good. I'm glad you asked that. This will definitely not be on the exam, only because we were in a rush and we never even talked about that yesterday. But what I do like about it is an important concept. The answer, and I'm just going to give it away since we didn't have time to talk about it yesterday, is B, reverse transcriptase. So HIV is an interesting virus because and I did get to say this yesterday, most viruses just give you the genetic information, whether it's RNA or DNA, and then your body just follows the instructions, right? Because that's all it is. It's a set of instructions. Going back to my analogy of your cell being a factory, a virus would be like someone just sneaking in, like the virus needs something to be made, right? So it just sneaks into the factory floor and just gives the directions to the machine. Remember the ribosomes, right? It's like, here, make this, right? And then it does. The machine just follows the directions and makes whatever you give it. So that's how a virus works, an RNA virus. A DNA virus would be more like sneaking into the factory, bypasses all the machines, and goes into the library where all the books of instructions are held, right? And it brings in the book of instructions and puts that in there, right? So it's the same concept, just that instead of going straight to the machine, it's adding the DNA, the original book of instructions, and then that is going to be transcribed, which will be translated. So that's how a normal virus works. HIV is very interesting. It's an RNA virus, and what happens is that RNA has this very unique enzyme called reverse transcriptase. And think about it. What is transcription? Transcription is going from DNA to RNA. So hopefully, logically, you can figure this out just by the name of it. Reverse transcriptase. This thing must do the reverse of transcription. And if transcription is from DNA to RNA, reverse of that must be RNA to DNA. And that is exactly what it is. So reverse transcriptase is an enzyme in the HIV virus that goes from RNA. It takes a strand of RNA, and from that, it makes a strand of DNA or a double strand, But that's the point. And then to me, the reason that's a slightly important concept is going back to the mRNA vaccine. Everything has its risks and its benefits. So I'm not going to tell you if it's safe or not, or let me reword this. I'm not going to tell you if it's worth the risk or not, because that is a personal decision. Some people don't think it's worth the risk to fly, even though flying is extremely safe. To them, it's just not safe. That's a personal opinion. Anyway, the thing to understand about the mRNA vaccine, now that we've talked about transcription, you should know it goes from DNA to RNA to protein, right? DNA to mRNA to protein. This mRNA vaccine skips the DNA part. So it just says, hey, here's the mRNA, build this protein. Meaning, and I've heard people say that, oh, this can mess with your genome. There's no way that this mRNA vaccine can mess with your genome, because it doesn't go from RNA to DNA, right? It goes from RNA to proteins. The only way it could go from RNA to DNA is if it had a very specific enzyme. And in the case of HIV, it's reverse transcriptase, right? It has the enzyme. So unless somebody put that into the vaccine, then it's not going to affect you. And no one's doing that. But anyway, sorry about the soapbox. Do you have another question? I just wanted to give you time to get your next question ready, O'Brien.
Still on chapter 10.
Okay.
43. Examine the genetic code table shown below. The codon AGC codes for the amino acid blank.
Okay. So here we go. We'll just do it. That's enough information right there. The code is AGC, right?
Mm-hmm.
Now, I just haven't decided if I'm going to have this on the exam or not. If you take the later exam, like if for some reason you slept in, which has happened, if you slept in and just missed the exam and you have to take it on a Saturday or Sunday or Monday, whatever, you're going to take a harder exam, and this will definitely be on there. But it's actually pretty easy. You start in the middle and go out. It's that easy. So look at this table right here, O'Brien. This chart. What would you say the first letter is?
A.
A. Good. All right. So then what's next?
A.
So 2 A's?
Yes.
Okay. And then what's next?
Then A.
No, I mean on the question you read me.
Oh, sorry.
That's all right.
A, G, C.
Perfect. So that's how you do it. You would start at the A, you would go to the G, then you would go to the C, and that leads you to the S. Oh, I think I might see the issue here. That would lead you to the amino acid that is abbreviated with an S. Hey, what in the world? Oh, I see the issue. Thanks, O'Brien. So I can see now that because I hit the shuffle button, that these things are not together. So what I looked for when I saw number 43, I searched for 43 and it brought me to this chart. It didn't bring me to the actual question. So let me go back to here and search for 43 again. All right. Thank you for letting me know. So now I'm gonna have to go in to everybody's study guides and then correct that, because if you missed that, I don't want to necessarily take that away from you, because that wasn't the way it was supposed to be. So yeah, AGC should be an S. And the only option here with an S is serine. So the answer is A, and that's how you would find it.
All
but
right.
more importantly, for the exam, I'll make sure, again, probably it might not have that anyway, but if it does, I'll make sure they're together and we don't have that issue. And that's how you do it. Yeah. So my apologies that those weren't together, but yes, it'll go any code. You start in the middle, whether it's an A, T, C, or G. You start with the first letter, then you go to the next letter, then you go to the next letter, and that'll tell you which amino acid you have. So, Brian, you're a rock star today. You're giving me a lot of information that I need, and you've been very helpful. All right. So what is next?
17.
Same exam, number 17, or same study guide, number 17?
Yes.
Okay. This is good. Transcription is the what? First of all, like I always say, I think the best way to study this is big picture first and then small picture. So that's one of the first big picture things you should know. We go from DNA to RNA to amino acids, or to proteins or polypeptides. DNA to RNA is transcription. RNA to proteins is translation. So let's focus on that. Transcription. Now that we know that, the reminder, that's DNA to RNA. Let's look at the options. The manufacturing of a protein based on information carried by RNA. Incorrect. That is translation. A, the manufacture of a strand of RNA, okay, complementary to the strand of DNA. Okay, so far A is the best option. Let me look through all of them. C, modification of a strand of RNA. Nope. Transcription, by the time they modify it, it's after transcription, right? That modification is between transcription and translation. That modification happens before it leaves the nucleus. Now you might, let's just leave it at that. So we know it's not that. B, manufacture of two new DNA double helices that are identical to an old DNA helix. So really, in my opinion, the only two close to being correct options are A or C, because you should know by now that transcription is not producing DNA. And I kept saying that too when I said you're going to get an exam question where it says transcribe this, and you can go ahead and cross out any of the options that have the letter T in it, right? Because there is no T in RNA, and you need to know that transcription, you're producing RNA. So really the only good options are A and C, because again, C happens kind of in between transcription and translation. So I could see where somebody might say that. As a matter of fact, when I write the exam, I think if you choose A, obviously you're correct. If you choose C, you'll lose some points. And if you choose anything else, you'll lose a lot of points, because at least C, it's close to it. Obviously it won't be labeled C in the exam, but you know what I mean. So the answer, transcription is the modification of transcription is the manufacture of a strand of RNA complementary to a strand of DNA. And when you're studying, keep in mind, I will probably change the words to this. So it might say something like, transcription is the manufacturing of a strand of mRNA complementary to a strand of DNA. Or I might just say transcription is the production of a strand of mRNA, right? There's so many ways to reword that sentence. Just make sure you understand the concept and don't memorize the words written in front of you. So any questions about that one?
No.
Okay, very important concept. Again, study the big picture before you add details. The biggest picture is DNA to RNA is transcription, RNA to protein is translation. Then start filling in the details. What are the three phases of transcription? What are the three phases of translation? Where does transcription start? Where does it end? Where does translation start? Where does it end? What are the parts involved in transcription? What are the parts involved in translation? Worry about all those details later. If you haven't memorized the big picture, then who cares about the details? That's a very important thing. again, going back to this idea of COVID and the vaccine, either one you're looking at, if you're going to do your own research, you need to understand these basics. Otherwise, you're just getting information that you don't understand, which is very dangerous. Not just for COVID, but for any concept. I just use COVID as the example because obviously we're in the middle of that pandemic. So, next question.
All right. Same study guide.
Okay.
Number 6. If adenine makes up 20% of the bases in a DNA double helix, what percent of the bases is guanine?
All right. I'm going to do another whiteboard. All right. You can see it, right? You can see my whiteboard?
Yes.
All right. So, we're talking about adenine, right? Is that what the first one they said? 20% is adenine?
Yeah.
Okay. What was the other one?
Guanine.
And what was its percentage?
It said what percent of the basis is guanine.
Oh, okay. So that's what we're trying to figure out. What percent is guanine? Perfect. So we know 20% is A. So what pairs with A? A is always paired with what other letter?
C.
C goes with G. So make sure you know this. Again, this is big picture stuff. So make sure you understand that before you even worry about this particular question. A always goes with T. T always goes with A. Very important. Very important. There's going to be a few questions where you need to know that. Plus, we've been talking about it since chapter 3. Well, not since then, but we first introduced it in chapter 3. A always goes with T. So every time there's an A, anywhere there's an A in your genome, there's a T. That being said, if 20% of your genome is an A, then what must T be? What percentage must T be? Because remember, for every A, there's a T. And there is no T without an A, and there is no A without a T. I'm hoping I'm giving you enough hints here. What if 20% of your genome is A, what must T be?
30.
Incorrect. Sorry, but I'm glad you're guessing. It's a 1-to-1 thing. A always has to be connected with T. For every A, there's a T. Meaning, if there's 20% is A, it has to be 20% T. Does that make sense? You can't have an A without a T, and you can't have a T without an A. Does that make sense?
So basically, it's going to be a 20 for A and T.
Yes. Anytime, whatever A is what T is going to be, and vice versa. Just like whatever G is, C is going to be, because it's a 1-to-1, right? They go together. So if I say G is 80%, and I'm not saying that, if G is 80%, then C must be 80%. Has to be, right? Because it's a 1-to-1 thing. You can't have an A without a T. You can't have a T without an A. You cannot have a G without a C. You cannot have a C without a G. So A will always equal T. Whoops. A will always equal T for the numbers. C will always equal G. Does that make sense?
Mm-hmm.
A very important concept. Okay. So we know A and T together, because now we know that A and T together was 20% plus 20%.
40.
Good. So we know 40% of this genome is made up of A's and T's. Therefore, what percent of the genome must be made up of G's and C's?
40%.
Here's what you have to think about. It's a percentage, right? So if 40% is A and T, then G and C, 40% plus this must equal 100%, because it all has to add up. So if 40% are A and T, and the whole thing has to equal 100%, then what must G and C be?
60%.
Perfect. Good. And I'm hoping this makes sense instead of me just giving you numbers. We already figured out why A and T altogether is 40%. And you know that it has to add up. A's and T's plus C's and G's have to equal 100%. So like you said, if A and T is 40%, then G and C must be 60%. Now, we know that A and T being 40%, because every A has a T and every T has an A, all you could do if you were working backwards is you could take that 40% and cut it in half, and then you would know, okay, 20% for A, 20% for T. Same thing could be said for G's and C's. If 60% of G's and C, if 60% are G's and C's, then what must be their individual percentage? What percentage?
30.
Yay, good, yes, perfect. Because 30%, if G is 30%, then C must be 30% because they all go together, and 30% plus 30% equals 60%. Very good. All right, you were asking like the best questions because these are for the other than the no, I'm not getting on to you or anything, but other than the transcription question, these are all ones where I actually have to explain it.
So A to T is always going to be 20, right?
Incorrect. That's just an example.
Okay.
A will always go with T. C will always go with G. I'm just going to throw out another number. Let's say A is 30%. So, or let's say the question says T is 30%. What is G? All right. Well, if T is 30%, then A must be 30%. Of course, this is kind of backwards of what we just did, but whatever. 30 plus 30 is 60. So 60% is A and T. Therefore, 40% must be C and G. Therefore, that must be 20 and 20. And the answer is 20. Point being, and this is a real life thing, when you look at a genome of a human being, they really do analyze it like this. Your numbers are not going to be the same as mine necessarily. There's a lot of, like, the genes are the same, as we've learned, but there's a lot of other DNA that's a little bit different. So it's not always going to be that example. That was just an example, and I'm so glad you asked that because I forgot to say it. For the exam, I'm going to use different numbers. I'm not going to use the 20. A is 20. I'm going to use different numbers, and I'm going to use different letters. Obviously, the letters are restricted to A, T, C, or G, but I'm not going to ask, I'm not going to give you A, and I'm not going to ask about G or whatever it was. I'm going to give you some other one. I'm going to ask about a different one, and the numbers will be different. But the concept is still the same. For every A, there's a T. For every T, there's an A. For every C, there's a G. And for every G, there's a C. Great question. You're doing all the ones that you actually have to figure out. So what's the next one?
All right. Same study guide.
Okay.
13.
Lucky number 13. All right.
What name is given to the collection of traits exhibited by an organism?
Yeah, this is one of those where, and I don't mean this in a bad way because you don't know it, but this is a good example of what I categorize as an easy question because this doesn't require you to figure anything out. It's just memorize it. This goes, just think about the lab, right? We looked at two things in the genetics lab. We looked at genotypes and phenotypes. Which one of them is the traits that are exhibited?
Phenotype and genotype.
Nope, just phenotype. You're right. You're right the first time. Because remember, the genotype is just your genes, your alleles. Do you have dominant alleles? Do you have recessive alleles? Do you have both? Right? That's your genotype. That's just your genes, your alleles. Your phenotype is the result of your genotype. So yeah, the answer is phenotype. Your genotype determines your phenotype. Your genotype is what alleles you have. Your phenotype is what's exhibited. It's the collection of traits, whether that be dimpled chin or sickle cell or achondroplasia or color blindness, whatever it is. Your phenotype is what is a result of your genotype. Obviously, genetic code is just thrown out there as just some random choice. Same with genome. And I'm really, again, thinking about this. I like the way I did the last exam with the point system because for this question, obviously it'll be reworded, but if you see this question on the exam, if you pick genotype or phenotype, if you pick phenotype, you're correct. If you pick genotype, at least you're in the right ballpark, so you're going to miss some points. If you pick anything else, you're going to miss more points. So, again, I'm trying to reward people who are studying. Clearly, you're studying. You happen to get that. I mean, you said phenotype or genotype, so at least you were in the right ballpark. And somebody who's not studying, they would probably, just as easily pick genetic code and have no idea why they were wrong. So, next question, please.
Number 7. In a double helix, adenine pairs with blank and guanine pairs with blank.
Good. And this is one that we just answered in a way, except we put numbers to it. So A goes with what, and G goes with what? So first, let's just stick with the first part. A goes with what?
T.
Perfect. And already we can see that that's the only option. So we know right off the bat that the answer is C. But to move forward, to let you know, obviously G, as we know now, pairs with C. So the answer is C. For the exam, again, I'm going to reword it. So it might not say air pairs with what and G pairs with what. It might say C pairs with what and T pairs with what, right? So it might be a little bit different. And the options might be a little bit harder because, like I said, like we just noticed here, right off the bat, we knew the answer was C because we know A pairs with T, and that's the only one that had a T. So you're going to have more options on the exam probably to make it just a little bit harder. And also, keep in mind that the only correct answers for this question can be cytosine, thiamine, adenine, or guanine, right? Because those are the 4 pairs. If you see any other word that's not a base pair of DNA, don't choose it because you're going to lose more points. If I throw in there a word like sister chromosome or, I don't know, some other random word from what we've been studying, you're going to lose more points. Your only options here for question 7 are DNA bases. Meaning this, D, you would have lost more points for D because uracil is found in RNA. It's not found in DNA. So you will lose more points for choosing that option as opposed to choosing any of the other ones. Whew. Next question, please.
All right. 39.
39. Let's see here. 39. A blank is to bacteria as a blank is to animal cells. This will also not be on the exam because we really didn't have time to talk about it. I mentioned it very quickly. Phage or phage, however you want to pronounce it, bacteriophage, phage for short. Those are the type of viruses that attack bacteria. And where we go here? Prophage. And a DNA virus is one of the types of viruses that attacks an animal cell. But again, I briefly mentioned that. So because of that, this will not be on the exam. But the answer is, I see. Wait a minute. Oh, excuse me. Let me look at this again. Okay. Antivirus bacteria, that wouldn't be it. Prophage. Yes, this is definitely not going to be on the exam. Because if I remember correctly, it's not even in the PowerPoint, even though the fact that we went through it quickly. So prophage is the type, that's when a virus attacks a bacteria. And instead of it just automatically happening, what happens is that viral genetic information is inserted into the bacterial genome, right? And a provirus is when the viral information, the genome of the virus, is inserted into the genome of the animal cells. So yeah, the answer is D, and it will definitely not be on exam. It was not, is not meant, definitely wasn't mentioned, and most likely wasn't even on the PowerPoint. I assume it's in the book. For 1 point of extra credit, if somebody can please tell me where in the book that is, or they talk about proviruses at least, then let me know. But anyway, next question, please. We only have about 20 minutes, so I want to help you out as much as possible.
All right. 19. Which of the following enzymes is responsible for RNA synthesis?
I like this. First of all, as we hopefully you remember from chapter 3 and or 5, an enzyme generally, if you're looking at the word, it ends with what letters? What suffix does it end with? And obviously you can look at the options and see what it is. That can give you a hint.
An R.
No, ACE. It always ends with ACE. Remember, like the first enzymes we talked about were ATP synthase, and the examples we gave in the lecture were lactase. Right? They always end with an ASE. So right off the bat, you should know that B is not the correct answer. Ribosome is not an enzyme. Right? It doesn't end with ACE. So for the exam, If you choose anything else, you'll lose some points. If you choose ribosome or something that's not an enzyme, you'll lose more points. So we know right off the bat, ribosome is not an option because it doesn't end with ACE, which tells us it's an enzyme. Now, we also said in chapters 3 and or 5 that enzymes are named either after their substrate or what they do. So if we remember back when we talked about cellular respiration, for example, the enzyme that makes ATP is called ATP synthase. It ends with ACE. It's named after what it does. It synthesizes ATP. Therefore, its name is ATP synthase. So based on these options, and we know now that ribosome is not one of the options, which of these things do you think makes a polymer of RNA?
Either RNA polymerase or DNA polymerase.
I could see that, but the answer is RNA polymerase. Because, like I said, it's named after what it does. So that's why I was trying to give you a hint with the way I asked it. What enzyme makes a polymer of RNA? The answer is RNA polymerase. I could see how that's tricky because you might think, okay, I'm hoping this is the way you're thinking because that means at least you were on the right track. Hopefully you were thinking, well, the enzyme that makes RNA attaches to DNA. It's reading the DNA code to make the RNA. I'm hoping that's what you were thinking because then you'd at least be on the right track. But remember, again, not just you, but anybody watching the video, first of all, enzymes end with an ACE, right? So anything that's not an ACE, throw it out the window. That's not an option. And enzymes are named for their substrate or what they do. So RNA synthesis, that would be RNA polymerase. It is the thing that makes an RNA polymer. Because remember, the monomer of an RNA is just that one nucleotide. The polymer is a bunch of nucleotides put together. So that would be an RNA polymer, right? So it is RNA synthase that does RNA. It is RNA polymerase that makes RNA. Likewise, for the exam, I might not ask about RNA. If it's about DNA, it's the same thing. Which of the following enzymes is responsible for DNA synthesis? Again, throw ribosome out the window because we know it's not an enzyme. So what enzyme makes DNA? The only option would be DNA polymerase because it's in the name. They're named after what they do or their substrate. This is such an important question. As easy as it is, because again, this is another one of those memorized things. You don't even have to figure it out. It's just a memorization thing. The reason this is so important is, one, you do need to know which enzymes produce RNA and which enzymes produce DNA. That's just something you should have memorized. That's one of the big basic topics. Not the big, it was medium-sized topics of that chapter. But also, like I said earlier, this incorporates information you learned in chapter 3 and chapter 5, and that has been reinforced every time we've talked about an enzyme, which is that enzymes end with ACE, and they're named after their substrates or what they do. So, great question. Next one, please.
31. Scientific research companies sell kits that allow researchers to produce proteins in test tubes via a process known as in vitro translation. Which of the following components is not needed in these kits?
I like this question. This is, again, one of those that I would categorize as a harder question. You can't look it up in the book. This requires you to use the knowledge that you've been given and put it to use. So we're talking about in vitro translation. So we're talking about translation here. Now we know, and we talked a lot about this when we talked about translation. Remember, before we even talked about the process of translation, we talked about the parts, right? So hopefully you remember the parts of translation are, well, first of all, you need the message that's going to be translated. That's your mRNA. You're going to need the machine that does the translating. That's the ribosome. You're going to need some amino acids, right? Because that's all translation is. All right, attach this amino acid to that amino acid to that amino acid, right? So you're going to need amino acids. How are you going to get the amino acids? Something needs to bring it to the ribosome. That's the tRNA. And then finally, of course, it's going to take energy to do all this. And your book says that it takes ATP to do that. So there we go. We know all the components needed for translation. Now, let's look at this list here. Forget this whole in vitro thing for now. Let's just look at this list. Ribosomes, yeah, we need that. Transfer RNA, yep, I listed that. That's the thing that brings the amino acids. Amino acids, yep, we need that. DNA, no, we do not directly need DNA for translation. Now, in your cells, we do, right? Because where does the mRNA even come from? The mRNA comes from reading the DNA, right? We know that. But if we're looking hyper-focused only on translation, those are the parts that we need for translation, right? DNA is not directly needed for translation. A good analogy might be when we're talking about cellular respiration, glucose is not directly needed for the citric acid cycle, right? Because what happened, what we had was the acetyl-CoA, right? Yes, the acetyl-CoA came from glucose, but glucose was not directly involved in that, right? Same in here. DNA is not directly involved in translation. So the answer is D, DNA. It's a great question. I would hope to include it because, again, it's one of those, not too many of these, but that's one that makes you think. You can't memorize this. You can't directly look it up in the book. Hopefully, if I include it, I would change the wording to where it wouldn't be like this, but I can't imagine how I could change the wording too much. But yeah, so there's the answer. Next question, please.
Chapter 9 study guide.
Okay.
Number 15. To determine the phenotype of an individual who expresses a dominant trait, you would cross that individual with an individual who blank.
Yeah, and this is not one of those, despite how long it is, this is not one of those questions where you need to figure something out. This is just in the book. And your book called it a test cross. And because we're running short on time, I would love to explain it to you, but I'd just rather help jog your memory. Look it up in the book. The answer is test cross, right? Or not the answer. A test cross. Like, all right, we have this individual who has the dominant trait, meaning we don't know if their genotype is homozygous dominant or heterozygous. So we used, I think, black labs as an example, or labs as an example. They were black, they were blind, whatever. And you could cross them. And what you would do is you would have to cross them with an individual who is homozygous recessive for the trait. That's the simple answer. And the reason being, really quickly, I can't talk about it too much. We're short on time. If you have an individual who's homozygous recessive, then you know when they mate with something else, the only allele that they're going to put out there is the recessive allele because that's all they have to give. That's why you wouldn't want to mate this person or this thing with a dominant something with a dominant trait or any kind of alleles that are dominant, because then you don't know what you're getting because the individual themselves are dominant. So that's all I can say about that. Look it up in the book. Go look at the video where we talked about it. We talk about it in great detail. Next question, please.
All right. Still chapter 9.
Okay. Oh, sorry, too. Let me say this really quick before you go there. For question 15, another thing you could do if you had the time, and some of you might, like if you asked me to take an exam one-on-one where I get rid of the time limit, then you could do this. Just do a Punnett Square for each of the possibilities, right? If the person, then that's exactly what I showed you to do in the video too. Like just do a bunch of Punnett Squares. But anyway, sorry. Next question. Chapter number 9, you said, or no, chapter 9.
Yes.
Okay. What number was it? I'm sorry.
Number 5. A purebred plant that produces yellow seeds is crossed with a purebred plant that produces green seeds. The F1 plant have yellow seeds. What is the expected phenotypic ratio of seed color of the offspring of an F1 multiply F1 cross?
Okay. Man, I wish I had more time to talk about this because this your book. When they wrote this question, they just tried to make it seem harder than it is. First of all, I want to remind you when it says F1, this right here, that's not a times. It's not F1 times F1.
Oh.
That's just showing that that's what's crossing, right? Like this mated with that. Sort of like if you had a yellow dog and a white dog mating, you would put yellow dog X white dog. In genetics, hopefully you've seen it a lot on the whiteboard or in the videos. When you see something like that, when we're talking about genetics, we're saying this thing mated with this thing. Anyway, this question just has a lot of superfluous words that are maybe just trying to confuse you. What this is really asking, it says, all right, a purebred mates with a purebred. And as we know, the purebreds are homozygous, right? So a purebred homozygous dominant mates with a purebred homozygous recessive. What are the results going to be? And like I said, but I said it in a different way. What I said many times in the lectures was, when we're talking about a monohybrid cross, which is this, when you're just looking at seed color in this case, one thing, one trait, this time or this mated with this, then you should expect a 1 to 2 to 1 ratio. And again, the other way to do this, if you had to, is just do the Punnett Square. Homozygous dominant crossed with a homozygous recessive. You should see that you would have the offspring in your little square. You would have 1 homozygous dominant, you would have 2 heterozygotes, and you would have 1 homozygous recessive. But again, shorthand, you should just know, like I said in the lectures, you should recognize a 1 to 2 to 1 ratio as a monohybrid cross. And what makes this tricky is I kept saying that. You should recognize it as a monohybrid cross. And this question doesn't specifically call it a monohybrid cross. It describes it. It describes a monohybrid cross, but it doesn't actually use those words and say, this is a monohybrid cross, which is great. Because again, this is one of those questions that slightly makes you think. Because not only do you have to know the answer is 1 to 2 to 1 for a monohybrid cross, you also have to recognize what a monohybrid cross is. So short answer is, the answer is A, 1 to 2 to 1 ratio, 1 homozygous dominant, 2 heterozygotes, 1 homozygous recessive. Next question.
All right, 32. Blank genes violate Mendel's principle of independent assortment.
This one I'll just give to you because we didn't really talk about it too much. And the answer is linked. Linked genes. Because remember, independent assortment means when all those chromosomes line up, it's not like all mom chromosomes on this side and all dad chromosomes on that side, right? It's random. So for all 23 of your chromosomes, when they line up, we don't know if it's going to be mom, and then dad, and then another mom and then dad, or maybe it'll be dad and then mom, whatever. It's random. So that's why things sort independently. That's why there is no correlation between chin dimple and widow's peak, because they're completely different. They have nothing to do with each other. However, if they're linked, that means they're on the same chromosome. Therefore, if you got your mom's, and I'm making this up, let's say freckles and eye color were on the same chromosome, that would make them linked. That would make them non-random. That means if you got your mom's eye alleles, then you probably got your mom's freckles alleles, because if they were on the same chromosome, and you're either getting your mom's chromosome or your dad's chromosome, right? So if you got your mom's, other than crossing over, then you got every single allele that she had for that chromosome. So sorry. The answer is linked. Next question, please.
16. A couple who has 2 female children, what is the probability that their next child will be male?
Okay, this one's interesting. I don't like this because I'm going I'll explain. This won't be on the exam. The way, by using the rules of probability, people can argue this, right? So technically, these are independent of each other. Technically, like it does every time you have a child, you're rolling the dice, so to speak, right? It's a 50% chance you're going to have a female, a 50% chance you're going to have a boy. Technically, right? Because technically, what you had prior to that doesn't technically matter. So by that logic, the answer would be B, right? But some people could say, sort of like rolling a dice. If you roll a dice, hopefully you'll know this, like hopefully you can remember. If you roll a dice, what are the chances of you rolling a 5, for example? Do you know the chances of that? Or let's make it easier. If you flip a coin, what are the chances of it landing on heads?
50%.
Yeah, it's either heads or tails, right? So technically, each time you do that, they don't affect each other. So in a way, every time you flip it, it's a 50% chance it's going to land on either one. But another way to look at it is the chances of that happening once is 50%. But the chances of having it happening again would be, the 1/2 times 1/2. That's the rules of probability. So then you could say it would be 25%. And the chances of it happening again a third time would be 12.5%, right? It just keeps getting smaller and smaller because by the rules of probability, you multiply. So if the chances are 50% of this and 50% of that, what are the chances of them both happening? Well, it's 1/2 times 1/2, which is 1/4, which is 25%. So that's why this question won't be on the exam, because you could argue it either way. Or maybe I will have it on the exam, and maybe I'll have both of those answers be correct, and anything else be worth. You'll lose more points for choosing anything else, and if you get either one of those, you'll get it correct. I don't know. We'll see, but that's how that works. For the rules of probability. Basically, all right, here's a better example. This is what your book was trying to ask. Let's say that there's a 1 in 4 chance of your child being born with a gene for bad breath. I'm just making that up. 1 in 4 chance of you and your whoever over there having a child with bad breath. 1 in 4 chance. So then the question might be, what are the chances of your child having bad breath, 1 in 4, and being a boy, which is 1 in 2, right? So 1/4 plus 1/2 is 1/8. So the chances of you having a child with bad breath, that's a boy. Those are two separate things. So for them to both happen, you would have to multiply the chances, and the answer would be 1/8. That's what your book was trying to get at. But in my opinion, that's a little too vague. So anyway, next question, please. Sorry to rush you. We're just short on time.
It's okay. 12. Which of these crosses will produce only heterozygous offspring?
This, we don't have time to talk about it, but in any situation like this, just do the Punnett Square, right? Do the Punnett Square for big A, little a. Do each one of those, just do the Punnett Square, and then find out which one produces only heterozygous offspring. And the answer is A. And I would show you, but we're short on time, so the answer is A. And again, just do the Punnett Square. And I know you're short on time, so anybody who wants more time to do the exam, today's the day to contact me, because tomorrow it's too late. So if you want to take more, like O'Brien, you said you're taking yours in person, right?
Yes.
So you'll have all the time. I don't want to say all the time, but if you go over your 50-minute allotted time, that's okay, because you're in person. I can see that you're not looking at a book, you're not looking at the internet, you're not getting help from anybody. I can't see that with anybody online. So anybody who wants more time, contact me today, and it still might be too late depending on what time you contact me or how busy I am today. So anyway, next question, please.
Let me just quickly find one before time is up. 36. Linked genes are usually blank.
This one won't be on the exam because we didn't talk about it. I mean, we did briefly talk about it, but I didn't talk a lot about it. But I kind of hinted at it with one of the other questions you asked me. The answer is D. They're located close together on a chromosome. Not only are they on the same chromosome, because like I was saying, the whole thing with freckles and eye color as a hypothetical example that doesn't exist. If they're on the same chromosome, like if you get your mom's freckle allele, then you probably got her eye allele because they're on the same chromosome. But even more so if they're close together, because of crossing over. The closer they are together on the chromosome, the more likely they are to travel together when you go through meiosis. So I have an idea. I am going to. You still have questions, right?
Only one more question I need to ask you. How many questions are going to be on the exam?
Good question, and I'll answer this. This will go for, well, it's the third exam, so I guess it doesn't matter anymore. It's going to be just like the other ones. I'm shooting for 50 questions total, give or take, and I'm shooting for it to be about, equal amounts, Chapter 8, Chapter 9, and Chapter 10. Now, what my plan was, because I do, like I said, I have to go, I have things to do, but what I can do, and this is up to you, I can join this meeting that we're having on my phone, so I can't display my screen anymore and I can't show you anything, but we can still talk. So if you have questions, you can still describe them to me, and I can't read them, so you have to do a good job of describing them. And then I'll have to try to verbally describe the answer if you want to do that. Otherwise, we can just end the meeting right now, whichever you want.
We can end the meeting now.
Can?
Yeah.
Okay. All right. Well, I hope this answered questions for you and anybody else. as always, if this didn't answer any questions you have, email me, let me know. Today's a very busy day for me. It really stinks considering that tomorrow's the exam, but I'll do my best to get to you. But it's a good time to remind you, don't wait until the end to study, right? Study these study guides. And if you have questions, come to me early. You don't have to wait until the exam review to do this. So good luck, everybody. And I guess that's it. Like I said, contact me if you need help. And O'Brien, extra credit. Oh, here's a good one. Let's do a 10% extra credit, meaning whatever score you get on your exam, O'Brien, you for sure, because you were here, and anybody who watched this video, you can get 10% of your exam as an extra credit. So if you score a 90, you get 9 points extra credit. If you score an 80, you get 8 points extra credit. If you score a 55, you're getting 5.5 points extra credit. To get this extra credit, you need to watch the video. Oh, let's make it a little bit harder to make sure everyone's paying attention. To get the extra credit, there's going to be a question. I'll put a question in on the exam that you will not know the answer to. So if you take a note, so Brian, do you have anything to take notes with?
I could put it on my phone.
Okay. I'm going to ask a question that makes absolutely no sense. And the correct answer is going to be flower pot. You got that?
Answer flower pot, right?
Correct. So when you see a question that makes absolutely no sense and you see flower pot as an option, answer it. That'll tell me, obviously you're good because I know you're here, but for anybody watching the video, that'll tell me not only did you watch the video, but you paid attention and you knew that part. So when you see a question that makes absolutely no sense on the exam, answer flower pot. And you will get a 10% extra credit bump to your exam, which kind of stinks now that I'm thinking about it, because probably the people who aren't watching this video are the people who are going to need the help. But then again, they're the people who should have been watching the video. So I don't know. That's what it is. Any questions?
No.
All right. I'm going to stop sharing, and I'm also going to stop recording here in a few minutes, but I don't want to do it right away because I don't want people to open up the video and skip right to the end just to see if I said anything about extra credit. So you can go if you want.
All right.
I'm going to continue to get ready for the day, put on my shoes and such, but I'm going to do it in front of the screen so it looks like I'm talking. We good?
All right. Bye.
All right, bye. Have a good day.
All right.
So I figured while we're still here anyway, if for some reason you got to the end of the video, not that you need to, sorry to disappoint if you were here hoping for some more extra credit or something like that. But let's see, could I offer any advice? Not really. Make sure you study the study guides. Hopefully you've seen from the past 2 exams that there's a correlation between those who take the study guide and those who don't. So please do it. Like I said earlier in the video, I'm very busy today. So unfortunately, if you need help, it might be hard to find me, but it wouldn't hurt to try. I will most likely be away from my computer. So if you need me, call my office, which is forwarded to my cell phone. That number is 304-766-3125. That's the only advice I can offer. Good luck, and I will see you Monday, unless you were one of those people who's already contacted me and will be here tomorrow, or not here, but be on campus tomorrow to take the exam.